everyone, welcome back to my channel Online Microbiology, and in today's class we are going to discuss about chromatography and its types. So if you see the chromatography, the word, so chroma means which is nothing but the color, and graphy means which is nothing but writing. So if you see the meaning of the word chromatography, so chroma means color and graphy means writing, so which is nothing but color writing. So the chromatography is useful for the separation, purification and testing. So if you see the chromatography, if you see the definition, it is useful for the separation, purification and testing of what means. For suppose if you take one mixture, the mixture will contain several compounds, right? So mixture is nothing but combination of several compounds. So if you want to study each compounds in a mixture individually, we know a technique called chromatography. So first it is useful for the separation, means for suppose I have taken one mixture. The mixture may contain proteins, it may contain carbohydrates, or it contains lipids, right? So this chromatography is useful for the separation of components in a mixture, and the second one is purification. So while separating the components in the mixture, we are also purifying the mixture. And testing means which is nothing but first separating the components and testing the components that are present in the mixture. So this is about the chromatography. So if you see the definition, so chromatography is a technique. So it is a technique that is useful for the separation, purification and testing of compounds. So in this what we can see is, so while studying about the chromatography and its types, first you need to understand these two words. What is the first one is stationary phase, and the second one is mobile phase. What are those two? First one is stationary phase and the second one is mobile phase. So by the names we can understand that stationary phase means the phase which remains constant, and mobile phase means the phase which will move, right? So in the chromatographic technique, whether it may be partition chromatography or gas chromatography or ion exchange chromatography, the entire working principle is based on these two phases. So stationary phase and the mobile phase. So what is the stationary phase and what is the mobile phase means, for suppose if you take the example of paper chromatography, in the paper chromatography we will take a paper, right? Chromatography paper. So this chromatography paper will act like a stationary phase. So means the chromatography paper do not move anywhere. So this paper is called as stationary phase. So actually if you study deep, the chromatography paper do not act. The cellulose contains water. That water will act as a stationary phase. This chromatography paper will act just like a solid support. So this is called a stationary phase. And while coming to the mobile phase, so this chromatography is placed in a chamber, and this chamber is filled with a solvent. So solvent means a substance in which a solute dissolves, right? So this solvent is called mobile phase. Now you can ask me, mobile phase means it will move, right? Then where it is moving? Means, so for suppose here we have taken one mixture, we need to separate. So what we will do is, which is the mixture we want to separate, that mixture will put as a dots in the chromatography paper. Now what will happen is, this solvent or the mobile phase will start moving up, right? So that is why it is called as mobile phase. Now how much distance the solvent will move the same distance, but the solute contains, means that material which we want to separate, it contains different compounds, right? So based on the solubility in the solvent, solutes will move at different heights. So these are the distance traveled by the solvent, and these are the distances traveled by the components. So by calculating that value, we can identify the components in a mixture. So in this about this we can study detail in later. First just try to understand stationary phase and the mobile phase. So stationary phase means in which the phase which remains constant. Mobile phase means solvent that moves on the stationary phase. So that is about the stationary phase and mobile phase. Now let us study about different types of chromatography in detail. Now what is the first type of chromatography is called liquid column chromatography. So what is the first type? Liquid column chromatography. So this is the first type of chromatography. It is called liquid column chromatography. So in this type of chromatography, what is the principle will be? So principle will be same. One stationary phase will be there, one mobile phase will be there. So in the liquid column chromatography, so one column will be there. It is made up of glass. So this glass column will act as a solid support. How it will act? It will act as a solid support, right? So this is the solid support. This glass column acts as a solid support. Now this glass column is filled with the stationary phase, which is the silica gel, right? So this silica gel acts as a stationary phase. I am keeping as a shortcut. SP means stationary phase. Now this silica gel acts as a stationary phase. Now what happens is the mixture to be separated is placed in the column. So this is the mixture which we want to separate. It contains different components, right? Now we want to separate. Now what will happen is, now stationary phase is there, we need to have mobile phase also, right? So this mobile phase, so this is the mobile phase. So this is the solvent, right? Now what will happen is, so during this process the solvent will move down, right? So while moving down, the solvent will also carry the sample particles down. So these sample particles will also move down. Now this these sample particles will be absorbed by the silica gel based on their absorptivity. Absorptivity means sticking to something, right? So for suppose, just consider this mixture has two samples. One is this one and one is this one. You just consider this one has more attachment power with the silica gel. So what will happen? Does it travel more distance? It do not travel. It will ultimately stick here, right? Now these components will have less absorption to the silica gel. Now they will not be absorbed fastly. Then they will travel up to here, and here they will be absorbed by the silica gel. So ultimately what will happen is, for suppose this mixture is a red color, you just think. So ultimately the samples are separated. So this will form blue color layer and this will form black color layer. So why? Because when both are combined, we'll get one mixture. When they are separated, they perform their individual characters. So by this what will happen? So the components in the mixture are separated based on the which principle? Adsorption or absorption, right? So that is adsorption, means sticking to the surface. So those samples which are having more adsorption power will travel less and absorb easily on the stationary phase. Those compounds which are having less adsorption, they travel more distance and they'll stick to the solid support or stationary phase. So based on this principle, what will happen? The components in the mixture are separated. So here what we have used? Column we have used, and liquid means the solvent and the sample is also a liquid. So silica gel is a solid. It is used column as a solid support. The components of a mixture are separated based on their adsorption. So this is about the first type of chromatography, which is the liquid column chromatography. Now what is the next type of chromatography is thin layer chromatography, right? So in this one what we are using is we are using thin layer. So which thin layer we are using means, so thin layer of stationary phase we'll use here, right? So thin layer of stationary phase means, so in the same way we'll take one glass plate. So in the column chromatography we have taken glass column. Here we are taking one glass plate. Right? So this glass plate is also known as chrome plate. This is the another name of solid support which we are using in thin layer of chromatography. Now this glass plate is coated with the silica gel which acts as a stationary phase. So silica or alumina gel. So this silica or alumina gel will act as a stationary phase in thin layer chromatography. Next, in the same way, we'll introduce now this chrome plate will introduce in the beaker like paper chromatography, and in this chrome plate 2 centimeters above the solution, above the solvent, we'll put one dot of the mixture which need to be separated. So below it was filled with the solvent. Now what will happen? The solvent will start rising up. Well, the solvent will reach the same distance, and these solute molecules will move based on their solubility in the solvent and the stationary phase. So, by measuring the distances traveled by the solutes or sample molecules, we can identify the components in a mixture and even we can separate. So, this is about the thin layer chromatography. It is also having one stationary phase and a mobile phase. In the column chromatography we introduced in the column. Here we are loading the sample on the glass plate which is nothing but the chrome plate. So, this is about the thin layer chromatography. And while going to the next one, which is the paper chromatography. While going to the next type of chromatography, which is a paper chromatography. I think you all know well about the paper chromatography. So it is very simple. All the chromatographies are very simple. You just need to understand what is the stationary phase, what is the mobile phase, and what is the procedure. Right? So in the paper chromatography, a paper strip is taken. So this paper strip will act as a stationary phase. So this paper strip is acts as a stationary phase, and this one is kept in a chamber that is filled with the solvent. So solvent will ultimately act as a mobile phase. I am writing the shortcut here, MP, mobile phase. So this is the stationary phase and this is the mobile phase. So in the thin layer chromatography, we have kept the chrome plate in a solvent, and we have kept the sample 2 centimeters, right? In the same way, we'll keep the sample 2 centimeters from the end of the paper strip. So here I have loaded the sample. Now this paper strip should be immersed in the solvent, right? Now completed our procedure. Stationary phase is there, mobile phase is there. Now this mobile phase will move on the paper strip by carrying the solvent. See, like this, right? While forming that one, it will also form the colors, right? So this is the principle under the paper chromatography. Now after some time, this paper strip has taken from the chamber and it is sprayed with the reagent. So then different compounds will form different colors. Then for suppose here we have achieved a green color, and later, so here also one sample moved and it has achieved blue color. Now what we'll do, we'll calculate the distance of this green pigment and this blue pigment, and we'll use one formula to calculate the RF value. So RF means retention factor. So how we can calculate means distance traveled by the solute. So solute is nothing but the sample, by distance traveled by the solvent. Now tell me, in the among the solute and solvent, which will travel the more distance? The solvent. Why? Because the solvent will reach until the end. The solute molecules only will move based on their adsorption. For suppose this is the chromatographic paper and this is the distance traveled by the solute and this is the distance traveled by the this is the distance traveled by the solvent and this is traveled by the solute. For suppose you just consider it is 3 centimeters and this is 1 centimeter. Now how we will calculate the RF value? RF is equals to, what is the distance traveled by the solute? 1 by distance traveled by the solvent 3, right? So ultimately the answer will be the RF value. So by comparing the RF values with the standard RF values, we can identify the components. So this is about the paper chromatography. And the next one is size exclusion chromatography, which can be also called as gel filtration. So I forgot to tell one thing, the paper chromatography is also an example of partition chromatography. What it is called? Partition. So partition is nothing but separation, right? So why it is called partition means, so here is the solvent is partition between the solute solvent and the water molecules present in the paper. So that is why it is called as partition chromatography. And while moving to the next one, size exclusion or gel filtration, in this, by seeing the name we can identify, here the components are separated based on their size and weight. So here the components are separated based on their adsorption on the stationary phase or solubility in the solvent and the stationary phase. So in the size exclusion or gel filtration chromatography, again one column will be taken, right? So this column will be filled with the semi-permeable gel beads. So semi-permeable means which allows only certain substances. Now what will happen is, now we have introduced the sample and we have introduced the solvent. So solvent is the mobile phase and this gel beads is the stationary phase. Solvent is the mobile phase. Now what will happen is, the components, the mixture will contain components of different sizes, right? So for suppose, if these particles are small, if these particles are small, the particles will enter the holes of these gel beads. So just assume the small particles has entered the gel beads, and whatever the remaining means, they will simply pass between the gel beads and they will come out. So which are larger than the gel beads, they are coming out. Which are smaller, they are entering into the gel beads. So by based on, first we will get the larger components of mixture and then we will get the smaller components. So by based on what? By based on their size, we are separating the components. So that is why it is called size exclusion or gel filtration, means here you are using the gel beads. That is why it is called gel filtration chromatography. So what is the next one is ion exchange chromatography and affinity chromatography. First let us learn about ion exchange chromatography. So ion means we can tell that which is having either positive charge or which is having negative charge. So for that separation, for the ion separation, we are using ion exchange chromatography. So here we are separating the components based on their charge, and they are eluted, means they are sent out using varying the concentration. Means for suppose here I have taken the solution that is containing positively charged ions and negatively charged ions. Now what we need to do is, in this we are having anion exchange resins. We are using anion exchange resins and cation exchange resins. Means what is mean by anion? Anion means negative charge. So if anything wants to attract the anion, it should be in positive charge, right? So here we are using anion exchange resin, means this resin will exchange with the anion. Now what happened is I have introduced this solution. Now what will happen? All the negatively charged ions will stick to it, right? All the negatively charged ions from the sample will stick to this positive charge. And do the positive charge and positive charge interact? No. So these positive charges will come out. So what will happen is, now all the negative charged ions are bounded to the anion exchange resin. Next, after that, we'll increase the salt concentration. So after increasing the salt concentration, what will happen? All the ions which are bounded to the resins will come out. So now first we have got the positive charge, and then later we got the negative charge. So by this what will happen? We separated the positive charged and negatively charged ions from the mixture. So that is why it is called ion exchange chromatography. Next, the affinity chromatography. So by the name indicates affinity. So affinity means interaction. So here we are separating the components based on their interaction with the ligand. So ligand means anything which is that is a substance which binds with the other substance. So here these resins will act as a solid support. Now we are having ligands. So just assume here we are having the sample. The samples are having star shaped components and the spherical shape. Now what will happen is only the spherical shaped ones can bind to these ligands. So when we introduce the sample, what will happen? All the spherical shaped ones will bind to the ligands, and the star shaped ones will cannot bind and they'll come out. Now we need to separate this also, right? Now what will happen is we use a solvent. So while using the solvent, what will happen? The solvent will ultimately come down, and along with the coming it will also carry the remaining components. So here we have separated the components based on the interaction with the ligand. So you use this process for the separation of antigens, antibiotics, antibody interactions. So for that we'll use affinity chromatography, right? So this is about the ion exchange chromatography and affinity chromatography. Still we are left with gas chromatography and high proficiency liquid chromatography. So what is the next one is gas chromatography. So in this gas chromatography we are using to separate the gas sample. So means components in the gas. So how means here we will be having one column. Just consider this is the column, and here in the column at the one end we will introduce the sample gas, means which we will not want to separate, right? Now this sample gas will pass through this column with the help of one carrier gas, right? So here the mobile phase is also a gas. So the sample gas is carried by the carrier gas, which might be mostly helium. Now this carrier gas will carry the sample gas, and the sample gas will move based on its physical and chemical properties. So it will move in the column based on its physical and chemical properties, and the end one detector will be there, and it will detect the sample gases based on the length they have moved. They will move based on their physical and the chemical properties. So that is the about gas chromatography. And what is the last one is high performance liquid chromatography, which is nothing but commonly called as HPLC, right? So in the high performance liquid chromatography, it is used to separate components which are less than 10 microns in size. So in the same way, here we are using sample. So the sample is mixed with the mobile phase. Sample is mixed with the mobile phase. Now here one pressure pump will be there. So we are using high pressure. This sample will be pumped into the column. So this by using the high pressure, this sample is pumped into the column. So this here mobile phase will be there. So this mobile phase will carry. So here the sample will again move based on their, as we discussed earlier, based on their adsorption. So which is having more adsorption, they will travel less distance. Which is having less adsorption, it will travel more distance. So they are inversely proportional. So this is the entire topic of chromatography and its types. So if you have any doubt, please comment below in the description box. And thanks for watching my channel, and please subscribe.